Hey everyone, and welcome to today's Lab Explainer. We're doing something really cool today. We're going to take a journey that connects the macroscopic world, a totally relatable everyday human reflex, straight down to the microscopic universe of culturing individual cells. If you've ever wondered how your body communicates with itself in just a fraction of a millisecond, and how scientists actually manage to study those communications in a lab, well, you are in exactly the right place. So let's dive right in. So, picture this scenario. What happens the exact second you accidentally touch a hot stove? Or maybe you're walking barefoot and you step right on a sharp thumbtack. You definitely don't just stand there, think about it for a second, and then consciously decide to move your hand or foot, right? No way. The reaction is totally visceral. It's immediate. You pull away before you're even fully aware of the pain. But how exactly does your body pull off that incredibly rapid reflex? Well, the answer lies in the specific routing of your nervous system. Take a look at your body's built-in warning system. Normally, a sensory signal has a pretty long distance to travel. It has to go all the way up to your brain to be processed before any reaction is triggered. That's the slow route. But for painful stimuli, your body actually uses an express highway. The signal takes a direct route right to the spinal cord, entirely bypassing the brain for that initial reaction. This super short distance allows for a rapid, involuntary reflex, which is what saves you from severe burns or injury. And the biological structures that make this express bypass possible, that's exactly what we're studying today. Here is our macro to micro protocol for the day. First, the hot stove reflex. Second, why scientists use mice. Third, prepping the glass cover slips. Fourth, extracting the DRGs. Fifth, culturing the pain cells. And finally, decoding pain fast. All right, part one, the hot stove reflex. The absolute star of our show today is the dorsal root ganglion, or DRG for short. You might also hear them called spinal ganglia or posterior root ganglia. So what actually are they? Put simply, within your peripheral nervous system, DRGs are clusters of cell bodies located right in the dorsal root of a spinal nerve. And their entire job? They act as the ultimate pain and touch messengers. The ganglion collects these sensory messages and transmits them incredibly quickly directly to the spinal cord. Basically, it's the DRG that makes pulling your hand away from that hot stove possible in the first place. Moving on to part two, why scientists use mice. Okay, so the really crucial point here is the staggering number, 98%. It turns out that 98% of human genes have a comparable matching gene in the mouse. So despite the tail and the whiskers, their DNA and gene expression are remarkably similar to ours. Because of this massive genetic overlap, and of course their small size and short generation times, mice are the absolute perfect, most commonly used animal model for studying human development and cellular functions, especially things like pain. Okay, let's get into the lab. Part 3, prepping the glass cover slips. You see, to culture cells, you really can't just use any random piece of glass. Our 25mm cover slips have to undergo a seriously rigorous chemical wash. First, they're immersed in a small beaker of acetone on a shaker at 70 to 120 RPM for 5 minutes. Then, they take a 5-minute spin in 70% ethyl alcohol, and that's followed by another 5 minutes in 99.9% ethyl alcohol. Next, we dry them using a Bunsen burner. Once they are totally sterile, we coat them by adding 500 microliters of poly-D-lysine polarate. You can kind of think of this as a molecular glue that helps the cells stick to the glass. Finally, we wash off the excess with sterile water, dry them under a UV light for 30 minutes, and let them incubate overnight before wrapping them up safely in parafilm. It is an incredibly meticulous process. Next up, part 4, extracting the DRGs. Over on our sterile prep bench, we rely on some highly specific surgical tools. Sure, scientists use standard scalpel handles and surgical blades, but they also rely on specialized gear. We're talking sharply curved Castor-Viho spring scissors and Vena's spring scissors that have a microscopic 2.5mm cutting edge. For some perspective, that is about the thickness of a quarter. All of this precision work happens over a dissector waxy plate using needles, Kim wipes, and Lox solution as our specialized liquid medium. The extraction procedure itself is basically a step-by-step masterclass in zooming in from the macro down to the micro. After the 2-month-old mouse is humanely euthanized and prepped, it's fixed over the waxy tray using 4 needles. The tissue surrounding the spine is cut away, and the spine is transected right at the level of the hind legs. Then, the upper part of the spine is cut off, and the spinal marrow is very carefully removed. Finally, using those super precise spring scissors and forceps we just talked about, scientists dissect the tiny DRG clusters. Keep in mind these things are barely larger than grains of sand. The goal is to collect around 30 DRGs from both sides. placing them directly into a prepared petri dish filled with 1,000 microliters of that log solution. But here is where the tension really ramps up in the lab. This is a strict warning. Scientists only have a 30 to 45-minute maximum extraction time. They are quite literally racing the clock to remove these glands before the delicate nervous tissue starts to degrade. It's a high-pressure environment for sure, where steady hands and speed are both absolute requirements. Which brings us to part 5, culturing the pain cells. First things first, the DRGs get a 5-minute treatment in a solution called Triple Express. While that's happening, we prep our highly precise digestion solution. This is 1 milliliter of N by medium with penicillin and streptomycin combined with exactly 20 microliters of a library's aliquot. That's just a perfectly measured microdroplet. We add a collagenase filter and this mixture into a tube, and then we transfer our freshly extracted DRGs right into this specific digestion bath. Now, this digestion phase follows a really strict chronological timeline to make sure the tissue breaks down properly without actually destroying the cells. First, we do a 7-minute digest in a 37 degrees Celsius water bath with some gentle shaking. Next, we use a micropipette to mix the solution exactly 10 times, then put it back in the water bath for 5 minutes. We mix it 10 times again, wait another 5 minutes, and then mix 15 times. Finally, to stop the enzymes from digesting too much of our sample, we halt the reaction by adding 100 microliters of FPS, and we just let it sit for a final 3 minutes before centrifugation. Okay, I have to enthusiastically warn you about a critical lab role during this timeline. During that mixing phase, which by the way scientists call trituration, the absolute most important part is that 0 bubbles are generated. Seriously, no bubbles. Why? Because if a bubble pops, the sheer force will literally tear our precious hard-won pain cells right apart. So the micropipette has to be set to less than 1,000 microliters, usually around 700, and the mixing needs to be done incredibly slowly and carefully. Once everything is safely mixed without any bubbles, we hit the final lab steps. We spin the cells down in a centrifuge for 4 minutes at 400 RCF to collect them at the bottom. We remove the leftover liquid on top, which is called the supernatant, and add 600 microliters of fresh NBA medium. We then seed these cells by carefully placing 100 microliters into each well right onto those beautifully prepped sterile glass cover slips we made earlier. Then they go safely into an incubator set to body temperature. That's 37 degrees Celsius with 5% CO2. After just 10 minutes of settling in, we flood each well with 2 milliliters of MBA complete growth medium. And there you have it, our cells are officially culturing. Finally, part 6, decoding pain fast. Let's look at the results. After just 24 hours in culture, we start to see initial DRG cell stabilization. You can actually see the DRG clusters starting to anchor to our chemical coating. But if you fast forward to 5 days in culture, or 5-DIV as the scientists call it, the difference is just massive. We see established, thriving cell growth stretching all the way out across the glass. The cells are healthy, they're fully networked, and they are ready to be studied. And that brings us to a final, really profound question to think about. How might studying these tiny cultured cells in a petri dish over 5 days unlock the entire future of human pain management? By taking that macroscopic hot stove reflex and isolating the exact cells responsible for it, using our genetically similar mouse models, scientists can now test brand new medicines directly on these living pain messengers. Understanding pain at this microscopic level, it's the first incredible step toward managing, or maybe even one day curing, chronic pain for millions of people. Thanks so much for joining me in the lab today for this explainer. Stay curious and keep exploring the science all around you.